So this morning we're starting with Donald Berry. Don is a professor in the Department of Biostatistics at the University of Texas MD Anderson Cancer Center. He received his PhD in biostatistics from Yale and had previously served in Minnesota and at Duke before going to the MD Anderson. He's written several books on biostatistics, has a large number of publications, and he's the principal investigator of numerous research grants from the NIH and the NSF. He's a fellow of the American Statistical Association and the Institute of Mathematical Statistics. And his topic this morning is revamping the RCT, including integrating observational studies. Don, welcome.
Thank you, John, and thanks to the program committee for inviting me. It's an honor for me to come and pay homage to a great person, somebody instrumental in evidence-based medicine. So I'm going to talk about randomization, randomized trial. Like all statisticians, I kneel at the altar of randomization. But I go down on only one knee. Randomization is not essential for learning. We wouldn't exist as a species if that were the case. Of course, not all of our specimens have benefited as much as others. Evidence is politics in the United States. In the words of Will Rogers, you can lead a man to Congress, but you cannot make him think. And if he were alive today, he wouldn't exclude female politicians from that if you ever look at the TV stations from the South. So I have to give a conflict statement. I'm owner of Berry Consultants, which designs clinical trials for a number of groups, certainly pharmaceutical companies, device companies, the NFL Players Association. I'm supposed to say how I mitigate the potential bias. One is by telling you about these biases, saying that they're real. My goals of improving the efficiency and ethics of clinical trials, providing companies with the associated methodology are the same that I used to discuss with clients, and that may attract clients to Berry Consultants. MD Anderson restricts my total income from Berry Consultants to 25% of my part-time salary, and of course by soliciting questions. Please ask questions at the question period. So I apologize for the bullets. Apparently the PowerPoint version that you have is different from mine. I'm going to talk about, I sat in the back yesterday and completely revamped my presentation. So some of the things I wanted to say I'm not going to be able to say, but I've added others that may be more germane. We've discussed the gradient of observational studies. Some are really garbage, some are incredibly useful, and I'll give you an example of that. And I want to focus on this media issue, which wasn't given much shrift yesterday. I'll tell you about adaptive randomization in a simple case and in a more complicated case. I'll say something about decision analysis, and I'm not going to be able to say much about the Bayesian hierarchical modeling for borrowing from observational studies into clinical trials. We've done that a good bit, actually, using, for example, historical controls and having randomization to control, but then using information that's available outside of the trial in a hierarchical way. It's enormously useful to take a Bayesian hierarchical approach. One views the various two levels of experimental units. One is the patient within the trial or the study, and the other is the study itself. And the historical controls may be similar to the controls in the trial, and you let the data tell you to what extent they are similar. It's completely prospective from the point of view of the trial. The bane is that it's not necessarily prospective from the aspect of using data to inform the trial. And there's where the thing that Salim mentioned yesterday about selection bias comes in. And so if a solution is to have everything be completely prospective, that is, choose which studies are going to be used to inform the randomized trial before you get the study data. That's not very practical, and so one has to worry about biases. So that's really all I'm going to say about that. This is an article that appeared a few weeks ago online. It's from the journal Cancer. It's in screening, and we talked about this a bit yesterday. In screening, it's incredibly difficult to have observational studies that make any sense at all. In breast cancer, for example, if you look at patients who were detected by screening as opposed to those that were detected by symptomatic clinical means, they're very different. They're even different all the way down to the stage, the Ki-67, the HER2 status, age of the patient. They're identical in every way. You might ask yourself, do you want to know how they were detected? And the answer is yes, with a vengeance. There's this stage shift, of course, but there's a within stage shift. There are many biases that come to the fore, lead time bias, of course, and length bias, perhaps first among them. This is an article that somehow made it into the literature. It's pure garbage. It's absolutely meaningless. The investigators from Harvard went into a database, looked at those patients who had died, and drew a conclusion that the patients were more likely to have died had they been unscreened. So, I mean, everybody knows that, as I just told you, that if you're detected by means other than screening, you have a much worse prognosis. That doesn't mean that screening is good. I once wrote an editorial called The Paradox of Screening. Better if found, perhaps better not to find. The problem is that the media picked it up. And this is typical. All of the news stations, the ABC, NBC, CBS, many of the newspapers, not some of them. New York Times and the Chicago Tribune missed out, purposely, I'm sure. They weren't duped by this. The Chicago Tribune wrote an editorial about 10 years ago where they were writing about another article, another piece of garbage in the journal Cancer. And what they said back then was absolutely germane for the current setting. We talked about millions of women waking up to news that the last word had been written in a debate over mammographic screening. Newspapers, etc., picked up, and the problem is that it's too bad it wasn't true. Once again, many in the media fell victim to well-intentioned authors of a new study without questioning the study's merits or calling an independent expert for a second opinion. Hey, it was published in a scientific journal, it must be right. The media is a huge problem. I mean, in a way, let people publish what they want to publish and let the user beware. But when the user is, when there's a middle person, the media, that's has different motivations, getting readership, promoting controversy, exploiting controversy, it's too bad. In case you're interested, this is the effective age. What the article says is maybe we should do away with some of the screening in the older women and put it in the younger women. In fact, the randomized trials say exactly the opposite. Essentially, all of the benefit of screening is carried by women in their 60s. So adaptive randomization, I want to start out with a very simple example. This is something we talked about yesterday. Only 1 in 5 oncology clinical trials are published. The biggest reason is they don't accrue. And one of the reasons they don't accrue is because they're not offering good things to patients. And one possible contribution to improving this is being adaptive. This is an article in JAMA about adaptive clinical trials, a partial remedy for the therapeutic misconception, which is that patients in trials think they're being treated as effectively as possible, whereas we know, the Belmont report says, that we do clinical trials to learn, not to treat patients in the trial. So this is an example. It's one of the first trials I designed when I got to MD Anderson. This is an experimental drug, troxacitabine, potential use in acute myelogenous leukemia. The endpoint of the trial was going to be complete remission at day 50, and it was a 3-arm trial randomized to standard therapy, idarubicin Ara-C, versus 2 other arms, experimental arms, which would include troxacitabine and either idarubicin or Ara-C. And the investigator said, I want to do a 75-patient trial, equally randomized. And I said, well, why don't we look at the data? And if the data are suggesting that one of the arms or another is doing better, let's emphasize it. And if it's doing sufficiently poorly, we'll drop it. There still is some element of randomization because as Susan Ellenberg indicated yesterday, we're randomizing patients to things that are looking not so good. There's no perfect answer to that dilemma, but perhaps this is a compromise. So we assigned with higher probability. After the 34th patient entered the trial, we had sufficient information to drop TI, troxacitabine, idarubicin. And it had been assigned to few patients, as you'll see, because it was doing poorly. That was after the 24th patient. After the 34th patient, the trial stopped when TA was doing sufficiently poorly. And these are the data. 34 patients, most of them were assigned to the standard therapy because it was doing much better. TI was doing very poorly, had 0 for 5 CRs. I don't know how to go back. That's all right. Thank you. Frank Giles, the PI, submitted the paper to the journal Blood, and the journal Blood said, the editor said, you can't do anything with 5 patients. And I said to Frank, let's send him a note saying, if he gets this disease, we have a treatment for him. But Frank was nicer than I was and submitted it instead to the journal Clinical Oncology. And they said it was clear that the drug is a dud, but the design minimized the number of patients who would be assigned to this treatment, and so we'll publish a paper. What's the benefit? So Susan said something about there are more patients being assigned to the trial. Not true, certainly not true in this case, 34 patients out of 75. If you compare the adaptive randomization to the balanced randomization, the adaptive randomization had this set of patients, 34 patients, with this 13 CRs. If you extrapolate to the rest of the 75 patients, who of course we get more than 75 patients who come into MD Anderson to get treated, the CR rate in that set of 75 patients would be 48% because, well, you know, that's a point estimate, because we had other, the other patients would be assigned to standard treatment. If you compare that to balanced randomization, it would have been 28%. If there were a drug that improved CR and AML by 20%, it would be a blockbuster. Frank Giles said there's no rationale to further delay moving these designs. He's currently involved in 8 Bayesian-based leukemia studies. They're more ethical, more patient-friendly, more conserving of resources, more statistically desirable. Problem is, maybe it's getting better over time, but the problem is what happened after. The company didn't believe it. By the way, I had nothing to do with the company. I was working through MD Anderson. The external impact of the study, the company built another trial. Phase 2/3 trial was never published. This is the only information I could find about it. The DSMB stopped the trial because it was killing patients. Completely predictable from our data, but the company and the investigators presumably didn't appreciate what the data showed. So it's a substantial ethical issue. I'm comfortable with what we did, but I'm worried about what the impact is in other circumstances. In 2006, the FDA's Critical Path Initiative was reformed to address what they call 2 issues: biomarker development and streamlining clinical trials. Janet Woodcock at that time suggested the improved utilization of Bayesian and adaptive methods could help resolve the low success rate and expensive phase 3 clinical trials. More recently, 2013, she said at a personalized medicine meeting, the FDA will need to turn the clinical paradigm on its head to allow personalized drug therapies to get on the market faster. And one of the things they've touted, and they've worked closely with us on, is I-SPY 2. I-SPY 2, they advertise it as by applying an innovative trial design, researchers then use data from one set of patients' treatments to decide treatment for future patients. That's adaptive randomization. This will help researchers learn more quickly which investigational drugs will be most beneficial for patients with certain biomarkers. So, Wall Street Journal, let me get to the cartoon. Oh, I have to comment on something yesterday. So Dr. Horowitz quoted Richard Peto to say, what's left, this is, the early clinical trials picked off the big fruit, and what's left for the rest of us is to detect small effects with large trials. My version, and Dr. Horowitz last night at dinner said he agreed, what's left, that's not true. What's left for the rest of us is to figure out who's benefiting from which therapies using small trials. And that's the whole point of I-SPY 2. If you could go back a couple, please, to the graph. We don't want to have 1,000 patient trials, which we are having in breast cancer in the adjuvant setting. We want small trials. So the cartoon, this is the way we usually do things. The population is regarded as being homogeneous. Consequences, 1 in 3 success rate in phase 3 trials in oncology. In I-SPY 2, we have many arms, many possible indications for the arms within the patient population. As we go, we might graduate experimental arm 2 into a focused phase 3 trial focused on this particular subset. Arm 3 might drop for futility. Arm 5 might graduate into a different subset. We add arms as we go. We've had 7 total experimental arms, close to 500 patients randomized, 5 different companies playing in the same sandbox with experimental therapies. And their offspring in various other cancers. Alzheimer's is a big deal. The I-SPY is being used in Alzheimer's. They've seen the writing on the wall. Last 20 trials, phase 3 trials in Alzheimer's have failed to the tune of billions, and a number of other indications. So I do want to say in 5 seconds something about decision analysis. We'd ought to, I'm not going to show you any slides on it, I'll just say, we'd ought to ask, why are we doing clinical trials? To benefit, to deliver better therapy, is my view. We'd ought to consider the patients in the trials, and if it's relapsed Wilms tumor, where there are 100 patients per year in this continent, we can't do 1,000 patient trials. It's impossible. We ought to ask the question, how do we treat this disease as effectively as possible? We want to learn. We want to take advantage of what we're learning. If we were to do a randomized trial with equal sample sizes in the 2 groups, it might be 10 total that would maximize the treatment of the patients in the trial. No power, but doing the right thing. And so if you get my slides, you can see the development of that. And I'll stop there. Thank you.